the steps to being the cure to cancer. What is a porphyrin for people that don't know?
Hemoglobin typically resides in red blood cells. And the reason why I bring up hemoglobin is because hemoglobin has the ability to transport oxygen from your lungs to the rest of your body. Proteins are not capable of transferring oxygen to the rest of your body. And so our biological system has engineered a way to create a unique molecule. Porphyrin resides in hemoglobin. And what porphyrin's capable of doing, porphyrin's capable of bringing in an oxygen and then transferring it throughout your entire body.
Welcome to Rarely Discussed, an American Porphyria Foundation podcast with your hosts, Andrew McManaman
and Nicole Castellano.
We have porphyria, a rare disease. Rare diseases differ greatly but share many similarities in how they affect our lives.
Being rare can feel lonely and isolating, but we're stronger together. Join us in meaningful interviews with porphyria and rare disease experts and the patients and families impacted as we connect, learn, and heal together.
We'll talk diagnosis, treatment, pain, mental health, managing daily life, and everything rare. Today's guest is Dr. Jonathan Arambula, a scientist, entrepreneur, and the CEO and co-founder of InnovoTEX, a biotech company that's redefining cancer treatment through a clinically de-risked drug delivery platform designed to precisely target aggressive treatment-resistant tumors. With a background spanning academic research and executive leadership, Dr. Arambula has built a career developing targeted therapies that bridge the gap between science and real-world patient impact. He previously held research and leadership roles at institutions like MD Anderson Cancer Center and the University of Texas at Austin, and is credited on numerous peer-reviewed publications and patents too. Now he leads the charge at InnovoTEX, where his team is pioneering platforms to deliver breakthrough oncology treatments. Dr. Arambula also lives with Erythropoietic Protoporphyria, EPP, and recently joined the Scientific Advisory Board of the American Porphyria Foundation, bringing both professional expertise and lived experience to the table. And today we'll explore how porphyrin-based innovation is shaping the future of cancer therapeutics and how personal insight can fuel scientific progress. So with all that being said, Dr. Arambula, welcome to Rarely Discussed.
Thank you. Yes, thank you so much for having me. That was a mouthful, by the way. So that was a nice intro. So I have to take that.
It was a tongue twister, but we talked a little bit before we hit record here, and just talking about this full circle and how things kind of justified in your path to your career and things like that. And chronologically, you talked about getting diagnosed with EPP and it kind of paving the way for you. So if you could rewind a little bit to diagnosis of EPP and how that kind of shaped your life today.
Yeah, no, absolutely. I think that probably a lot of other porphyria patients will have a very similar experience to mine. So I don't think I'm a unique snowflake by any stretch of the imagination, but I do, remember, at 4 or 5 years old, getting some sort of exposure, for the very first time, and my parents not really knowing what to do. They see, a kid in pain or not doing well. The first thing they do, first inclination, take them to the pediatrician. Pediatrician just kind of does a surface type of assessment. And I remember continually getting diagnosed with just severe allergies to pollen, plants, whatever it is. And so this happened for several years. And I remember one time, one of my exposures was so bad that, I started to blister and bleed and, all over my face and my hands. And then my parents then took me to a dermatologist who eventually kind of remembers one of those weird, rare diseases that they learned about in medical school and kind of put 2 and 2 together. And so around age 7, I was officially diagnosed with EPP. And I remember, like, as a 7-year-old, I can now remember things. And so I remember as a 7-year-old, getting this diagnosis and having a certain sense of relief, essentially knowing what my problem was. And I remember the dermatologist basically saying, well, do your best to stay out of the sun. And I was like, really, I'm a 7-year-old. Like, I want to go out inside and play. I want to throw the ball around. I want to just, play with my friends. And I couldn't do that. And as time progressed, I started to realize more and more that there really was no solution to my condition, this rare disease that nobody talked about. I was the only person. I grew up in El Paso, Texas, which is arid desert climate, right? And so,
Yeah.
not the ideal conditions for a new kid diagnosed with EPP. And it was very disheartening for me that there was really no solution other than to just stay indoors. And that really took a toll on how I kind of, viewed the world moving forward. But it also guided me. Like, as I'm older now, so I can kind of come to terms with a lot of this stuff. But it really did guide me. As a child, being told that there is no solution for something you were diagnosed with was really, really impactful. And I do think that shaped my career choices and my path and my desires and my interests as I moved into college, as I moved into graduate school. As I became the person that I am now in terms of my mission statement, so to speak. And so I remember kind of just going fast forward a couple of years. I go into undergrad, and I was pre-med track, because I didn't really know anything else. I was on that track, doing fine. And then my organic chemistry professor came in one day and he said, "Shut your textbooks." And he basically said, "Let's talk about some real world applications with what we're learning today." And he showed us that in his research, he was actually making experimental drugs for cancer, for oncology. And that was kind of like that aha moment for myself, because I found real world application that I've been seeking so desperately in a type of science that I really did enjoy. And so I remember essentially that day I dropped pre-med, I joined his lab, I got, I changed my major to chemistry, and the rest is sort of history, because chemistry is one of those, it's physical science, but yet it also teeters on the edge of biology. It's kind of at the interface of a lot of different fields. And you can apply it towards creating life-changing therapeutics that could actually change patients' lives. And that was mind-blowing to me. And that's really that's really the thing that I wanted to devote my life to. And so, I think about this, like you said, full circle, EPP diagnosis. It really did shape a lot of my decisions moving forward, because I thought that with the right education, I could create something that could be impactful for patients also suffering from diseases that don't necessarily have an immediate cure. One perfect example being cancer. Mm-hmm. So, I am a firm believer in the sense that, even though EPP does have its severe limitations, on a day-to-day basis, I do think that I have been able to take something positive from my experiences and and those positive experiences have really influenced my decision making into my professional career.
Yeah, and I think leaning into that circle a little bit more, and I know talking to patients, I mean, even since this podcast came to be, there's just their biggest complaint is just the lack of empathy. and from doctors when they're
Misunderstanding, lack of education.
Mhm.
Absolutely. How many times have I gone to a family event or, where there are people who, they're kind of on, they're not on the outside, but they're sort of,
Mhm.
on the inside. You tell them about your disease and you tell them about your condition. And so many times I've heard people say, "Oh, I burn easily too." Or, "Oh,
Mm.
I wish I had that because I don't like the sun either."
Right.
And so I'm sure everybody's got that story. Um,
Yeah.
but yeah, yes, I think it really boils down to a lack of education, a lack of awareness regarding diseases that people do suffer from that are not your run of the mill type of, epidemic, such as, if I had a skin cancer, for example, or melanoma, and I had to stay out of the sun. Because of that, people would be a lot more empathetic or understanding because that's something that they could potentially suffer from as well. And there's a lot more awareness. So I would say that with respect to the porphyrias, there is an absolute lack of awareness, a lack of education because it is a rare disease.
Mhm.
But because of those things, there is a lack of empathy. You know, um, I don't think anybody is being, sinister. It's just a lack of understanding.
Right. Right. And I think just to nail that home some more, I mean, to hype you up a little bit, I mean, since you do have EPP, you have that
Yes,
built-in empathy for those people that are going through all these, whether it's the cancer aspect, I mean, you can feel
and
that
yeah,
to some extent. So I
I
guess
think it helps. I yes, I think it helps. It's easier for me to draw a connection to the person suffering from the the disease or whatever it is. So I do believe that there's a stronger connection. I do believe that there is a stronger sense of empathy that I have with respect to that, and I think that that really helped fuel my academic endeavors. I mean, I started as a professor, and I was really driven by this idea that I wanted to help people. Um, and so that I think that is fueled by a higher level of empathy for those individuals.
for
So
sure. And with that being said, too, just how it's paved the way for your path and obviously creating the businesses and things like that. What
mhm.
what made you on your path choose the business that you did in terms of the my mouthful that I spewed earlier about just the therapeutics regarding cancer and things like that. What made you choose that path?
Right. So fast forwarding, I think I ended at undergraduate. It's based on
Yeah.
my life experience. Fast forward, I always wanted to be a professor. I wanted to teach. I wanted to educate. I wanted to have a lab where I could innovate and use my kind of creativity in creating things that could be of utility for a general population in a translational way. I've always been very dedicated to translational science from benchtop to the clinic. That's always been a strong guiding force in how I think about things and how I operate. And so I took a position as a professor at Georgia Southern University, and I was teaching students. I loved it. I thought it was great. I taught biochemistry, had my cancer lab, doing cancer research, chemistry as well. And during that time, when I was as a professor, there was a fork that was kind of created in the road for myself. So my dad was diagnosed with a grade 4 glioblastoma. And so glioblastoma is a type of very aggressive brain cancer, and it's grade 4. So it grew rather quickly. Um, in hindsight, we could see the signals, but we thought it was just ocular degeneration. But he was diagnosed, And as soon as he was diagnosed, we knew that the clock was ticking because the survival rate on this type of cancer is essentially zero, a 0% or a 5-year survival rate. And he was in El Paso, Texas, where we grew up, but we knew that there were some more advanced medical centers over in East Texas, kind of like where the majority of the population is: San Antonio, Austin, Dallas, Houston, all of that.
Mhm.
And so it created sort of a fork in the road for myself because I knew that I needed to get back to Texas to be able to get to an area to be able to provide him with some, a little bit more robust care in certain parts of the country. And I also wanted to spend more time with him because the clock was ticking. But from a professional standpoint, it also created a fork in the road for myself because I was at the university, I was teaching, focused on education, focused on innovation that was very, very early. And it could not, the chances of early stage innovation, the likelihood of those ideas actually turning into life-changing technologies, there's a very slim chance. I mean, the odds are against those types of technologies. And so, but my postdoctoral work that I had completed, I had a staff position as well at the university. And that technology that I had invented years ago had been incubating and developing and kind of fostering in a very positive way to where that could actually be something that was potentially life-changing. And so I decided, I made the hard decision to leave my academic position, move back to Texas, kind of spend the last time, with my father. And there was an opportunity to start turning university technologies into companies, kind of spinning them out from the university and kind of moving into that early stage setting. And so and I go back to one of my themes. I've always been interested in translational science, moving something from creation to the clinic. And so this was kind of getting back to that kind of guiding principle that I've always been very interested in translational science. So we started a company in 2016, late 15, I believe. Started my first company with my old postdoctoral mentor, Dr. Jonathan Sessler, which is a key component in this whole journey to date. So,
Gotcha. And something I'm just because I don't have the knowledge of everything that goes into something like this, because you talk about the creative side, and in a sense, there's that experimental aspect. I mean, what kind of hurdles do you face taking it from what, like you said, creative to clinic?
oh gosh, that's a really good question, and that's a question that I oftentimes talk to a lot of graduate students about, even to this day. Um, not everything, not every wonderful scientific idea has translational value. And there's a lot of different things to consider, not just from an innovation perspective, but also protecting an idea, de-risking an idea, um, being able to conduct the right experiments, to be able to um, create a data set that justifies its potential in the larger marketplace. There's a lot of different factors to consider in moving a early stage innovation from a university into an actual um, corporate type of setting. But I think, if I were going to kind of give a blueprint to a young graduate student or a young postdoctoral fellow or somebody who really wants to formulate this idea is, you know, I would obviously early experimentation to show its potential, is absolutely essential. Um, so whatever experiments you would have to set up to be able to show that your idea is better than what else is out there, but also having a good understanding of the market, what else is out there, and how many patients could you potentially treat, and not to talk about the seedy underbelly of the pharmaceutical industry, but how much, what's the market, how much could be potentially made, because you're always going to need investors, and investors oftentimes need to make a return in some way, shape, or form, so that also has to be considered. And also, is your idea patentable? I think that's one big thing that a lot of early graduate students don't necessarily consider or think about, is whether or not your idea is innovative enough um, to be able to file some protection around it, is another consideration.
And I would imagine there's just so many ebbs and flows to all of that. I mean, you have something you work so hard on that just doesn't go anywhere. And just to your point and to your research, I mean, how are you able to break through that and just wake up every morning and just say, I just got to keep going, got to keep pushing through this?
The ebbs and flows, I think, is an interesting comment, because one um, lesson that my postdoctoral mentor, Jonathan Sessler, now business partner, always tells people is, don't be too in love with your own technology, because we can be, as scientists, we can get we can fall too much in love with our technology, thinking that it is really just the most wonderful thing. Um, and so we have to keep some type of perspective with respect to whether or not the technology we are developing actually has a fit for purpose within the larger market. What was the second part of your question? I'm sorry, I totally forgot.
No, you're good. Just the aspect of just doing your personal research and being able to deal with
Mhm.
those ups and downs and what motivates you to keep going in that
Yes.
regard.
Yeah. Um, Belief that your technology has potential. Once you get to a certain level of de-risking and you see the potential, you see the market, you see the opportunity, I think having that belief and being able to effectively convey your story to not just investors, but just the general public. From even it, it could be an investor pitch, it could be just an educational opportunity. Being able to effectively tell your story in a clear and concise way to captivate an audience is really, really key to this process. And that's something that we've spent a lot of time on over the last 3 years since we incorporated InnovoTEX. Um, and it's always good. I will say it is easier when you have people that believe in you.
Mm-hmm.
And we have a really great set of early stage investors in our company who absolutely believe that we have great potential, and we have a great fit. We have a great we can make a significant difference in the therapeutic community for oncology purposes. And so I think having that um, I think having people like them make it easier to get up in the morning and keep charging along. And, whether it's a scientific meeting or a meeting devoted to development, or whether it's a meeting with a new potential investor, you know, it that's one thing that definitely makes it easier.
And with motivation and discipline in mind, would you say that what your dad was going through is something that motivated what you're doing now in terms of cancer therapeutics, or was that just a coincidence?
No, I think it's an absolute motivator.
Okay.
Yeah, no, absolutely. I saw what he went through. I saw the good days, I saw the bad days, and I saw the toll that the medication actually took on him.
Mhm.
I saw, the surgical procedures, the resection of the brain tumor and everything. I saw every single aspect of what he had to go through, and it's not a comforting thing to experience. Unfortunately, it's something that hundreds of thousands of people in the US alone experience on a daily basis. And, you know, I I feel for them. It's a terrible thing to experience. And I absolutely think that that is a massive motivator as to why I believe our technology has so much potential from a safety standpoint, from an efficacy standpoint, and so forth. And that's why that even further drives me to get this technology into the clinic so we can start seeing those types of beneficial responses that we expect to see.
Right. And when I was talking to Desiree about
Yeah.
potentially having you on the podcast, and what was intriguing to me, because I unfortunately have the personal experience from cancer. My dad passed away with lung cancer,
Oh, I'm sorry.
and seeing the deterioration of that, and like you said, the medications, the chemo, the immunotherapy, all the stuff that he had to go through, and was not. It just did the opposite of what it was supposed to do.
Hmm.
And of course, everybody's different. It just depends. But what was intriguing to me is when she brought up the porphyrin aspect. And
Right.
I think for the people watching, I know, or listening, we talk about porphyrins, it relates to porphyria, but just for people who have no idea what's going on with porphyrins and porphyria and just how that ties in, and of course how it ties into your therapeutics, what is a porphyrin for people that don't know?
Porphyrin it's okay. So most people, I hope, have heard of hemoglobin in the body. Hemoglobin is a protein, and hemoglobin typically resides in red blood cells. And the reason why I bring up hemoglobin is because hemoglobin has the ability to transport oxygen from your lungs to the rest of your body. We can all appreciate the fact that oxygen is essential, in the survival, and all of our cells in our body require oxygen in order to survive. Now, the big question is, and sorry if I'm getting too professorial,
No,
but
you're good.
one, proteins are not capable of transferring oxygen to the rest of your body. And so our biological system has engineered a way to create a unique molecule called a porphyrin. And a porphyrin resides in hemoglobin. And what porphyrin's capable of bringing in an oxygen and then transferring it throughout your entire body. So it's the transporter for oxygen. So porphyrin is actually an essential part of our biological system that allows and facilitates survival and growth and just everything that we need, because our body is entirely dependent on oxygen for survival. So, porphyrins have a very valuable role to play. So, once we start getting some sort of metabolic disruption, in the formation of porphyrins. That's when we get all these different types of porphyrias. And so they can cause problems when they end up where they shouldn't be ending up, is kind of the simplistic way to put it. So porphyrins have a valuable role to play. And so think of a porphyrin as a donut. This is a circular molecule, has a hole in the middle. And that hole in the middle is really important for housing or holding a metal. It's kind of like the jelly inside the donut. Can't think of anything else. And so that metal is actually important. So in protoporphyrin, it houses an iron, and that iron is really important for holding oxygen and transferring it to where it's supposed to go. So the story of our technology and the use and the observations on how porphyrins could be utilized for cancer treatment started years ago in my postdoctoral advisor's lab, Jonathan Sessler. This is back in the 80s, I believe, and he had noticed a couple of interesting observations with just general porphyrins. In that general porphyrins, if you took a tumor out of a body and you looked inside of it, you would notice a ton of porphyrin just building up and collecting inside the tumor. And if you look at the literature, if you do a Google search, if you do a ChatGPT search, and you say, does porphyrin localize to solid tumors, you'll get all of this information showing how porphyrins themselves tend to aggregate and localize and get retained in solid tumors. There's a variety of different reasons behind it. I won't go into those. But it was an interesting observation on his part. And so he thought to himself, well, what if we took a porphyrin, the donut, and just made it bigger? If we made it bigger, we can put different things inside of it besides just iron, and we can put metals that have unique properties. And so that's what he did. This was actually his tenure work. So he got tenure at the University of Texas at Austin for doing this. And he found that these expanded porphyrins could have unique metals that had very different properties. So one of them that he made was MRI active. So you could actually see it, visualize it using an MRI. You could actually see and visualize it going to different parts of the body. And when you make the porphyrins bigger, they have also other properties as well that had potential therapeutic value for oncology purposes. So he starts a company as soon as they discovered this. He starts a company called Pharmacyclics, or he co-founds it with one other individual, Richard Miller. And they start to develop these expanded porphyrins for various purposes. One was for arteriosclerotic plaques, or just plaques that build up in the legs and so forth. And another one was for oncology. They ended up focusing on oncology. And this was developed for metastatic lung cancer, so lung cancer that metastasized to the brain. In the early phase 1, phase 2 trials, they found that this expanded porphyrin localized very, very well to all kinds of solid tumors. It went to solid tumors and it retained solid tumors. You name it, head and neck, lung, colorectal, the number of solid tumors that it localized to was just immense. They eventually had to pick a cancer type to focus on for the phase 3 trial, so that's what they did, which was metastatic lung cancer. And so they developed it as a radiation or an adjuvant. So what an adjuvant is kind of like a combination therapy with radiation. So they would give you the Texiferin. Give you the Texiferin. It would go to the tumor. They would then shoot the tumor with radiation. And then when the taxiferin was inside the in the tumor, it would basically do what normal porphyrin does to an EPP patient. So there's kind of some crossover, creating ROS species, creating oxidative stress, creating free radicals that damage and destroy the tumor. That is exacerbated by that radiation. So that was the whole scientific premise behind its original development, and it worked great. It worked great in so many different patients. There were for the phase 3 trial, it was working well. Unfortunately, there were a couple of sites in France that took a very different standard of care protocol. So what they essentially did is they put patients on a trial, then they let the patient sit for a couple of months and then get their treatment. And so this is obviously problematic because you're treating metastatic lung cancer. The patients don't have months, right? And so it's that those sites, they skewed the statistics. It's all published. So in the US, they absolutely met their endpoints. Globally, like if you look at all the data, they did not meet the statistical endpoint for overall survival. And so this was during a time when the FDA did not look at individual data, like from the US and stuff like that. So ultimately, it did not receive commercialization or FDA approval. But it worked. In general, it worked very, very well. But one of the key things that needs to be emphasized from this expanded porphyrin is that it localized very, very well and was highly, highly retained in every single solid tumor evaluated. I mean, that is the one key observation from all the clinical trials of over 1,000 different patients that's really undisputed. It was its ability to localize. So at that stage, the technology was returned to the University of Texas as the inventing institution. So UT adopted all of the IP surrounding this. So if I were talking to an early graduate student, really consider your IP. The university adopted all of this IP and has been maintaining it. And so all the technology that really Pharmacyclics basically had all the intellectual property for these types of expanded porphyrins. And so when the University of Texas received all of that IP, it really gave the university kind of this freedom to operate and kind of explore new paths or new applications for this technology on an asset that's already been clinically de-risked. I mean, one thing I forgot to mention about this technology, it was very well tolerated by patients. So patients in an oncology setting didn't have any grade 3 or grade 4 adverse effects or adverse events. And from an oncology therapeutics perspective, that's very good tolerability at daily dosing as well. So it was, the 3 take-home messages for my long-winded story is it was very well tolerated. It was a very good tumor localizing agent, and you could visualize it. Remember, this is MRI active, so you can see it in real time actually going to solid tumors. Like, what other drug can do that?
Right.
And so these were the 3 take-home value adds that the university and the Sessler lab had identified, and they wanted to find an application for it. And so this is around the time of 2008, 2009. I'm young, naive, wanted to work in translational science, and I was always very interested in Jonathan Sessler's lab. And I finished graduate school around that time at the University of Illinois, and he I was able to join his lab as a postdoctoral fellow. And so eventually I received an American Cancer Society postdoctoral fellowship for my time and tenure at the University of Texas at Austin and MD Anderson Cancer Center. But I came in and we had this crazy idea of taking this porphyrin, this big porphyrin that's very well tolerated, good tumor localizing agent, and you could see it, taking this big thing and attaching therapies or anti-cancer therapies to it. So the whole premise or the whole thesis or hypothesis that we had was, if this thing's very well tolerated and goes directly to solid tumors, why can't this expanded porphyrin then drag chemotherapy, for example, or a targeted therapy or whatever, a therapy? Why can it not drag that therapy directly to the solid tumor?
Hmm.
So you have to think about the market. Okay, so let me just take a very brief step back, and you have to think about oncology or just therapeutic treatment. Solid tumors can be divided into two different types: solid, and then we call them liquid tumors, which is essentially like leukemias and stuff, like blood cancers. And so, in a variety of different solid tumor types, you get diagnosed, and then you get put on some sort of anti-cancer regimen. More often than not, it's going to be some type of chemotherapy or chemotherapy cocktail. Likely in the context of lung cancer, like your father, is probably a chemotherapy plus an immunotherapy or some sort of combination thereof. But more often than not, patients are going to receive some type of chemotherapy. Okay, so chemotherapies are an interesting beast in the sense that they have saved more lives than, I'm pretty confident in saying that chemotherapies have probably saved more lives than any other therapies combined. They've been around since the beginning of time, well, 50, 60 years.
Yeah.
And most people will receive some sort of chemotherapeutic treatment as the standard of care for whatever solid tumor indication. Now, depending on indication, so I don't want to speak for all different cancer types, but in general, it will work initially and then it won't work eventually. Or it will work initially and then patients have to come off of the treatment because of the side effects. The reason why this happens oftentimes is most drugs, targeted therapies, chemotherapies, immunotherapies, antibodies, you inject them into the body. Initially, they go everywhere. You can't fight the second law of thermodynamics. They will go everywhere. Eventually, certain drugs will stick to the tumor. Antibodies that are targeting something specific on the tumor, for example. But chemotherapies, they tend to just go everywhere. You expose your entire body to them, hoping that the tumor will die faster than the rest of your healthy tissue.
Mm-hmm.
And so patients suffer from a lot of different side effects. Okay, so there's a side effect issue. There's off-target events that need to be considered for a variety of different treatments, in particular chemotherapies. Now, chemotherapies eventually will stop. The tumor will stop responding to those chemotherapies. And the most common chemotherapy that's used are platinum drugs. And so platinum comes in a few different forms, but ultimately what will happen is that patients will eventually build up resistance to these platinum drugs. And in oncology, as opposed to all other disease types, you give as much drug as a patient can possibly handle, hoping to kill the cancer.
Mm.
So you oftentimes will treat at the, maximum dose feasible for that individual or that patient, because you just want to eradicate the tumor. So when a patient, when a tumor builds up resistance and the drug no longer responds, you can't dose any higher, because the
Right.
more you dose, they're just going to suffer from side effects.
Mhm.
They're just going to suffer from these dose limiting toxicities. They're going to get, irreversible, kidney damage or nephrotoxicity. They'll have peripheral neuropathy, myelosuppression. They suffer from a variety of different, depending on the disease and the type of chemotherapy that they're receiving. So you can't just dose higher. It's just not an option. So then you have to go to second, third line therapies. And a lot of cancer types, the second and third line therapies, I mean, they're marginal responses at best. That's assuming that, that's just, the patient population that actually responds. So we need better options. We need safer options. We need options that can deliver treatments to the tumor. And so when I started graduate school, it's coming full circle again. So when I
Yeah.
started when I started my postdoctoral fellowship, we had this idea: take something that was well tolerated, not a lot of side effects. That's a win.
Mhm.
And attach chemotherapies to it to be able to deliver it to the tumor. There was this belief that if you could increase certain chemotherapies by just twofold in certain tumors, like if you could get just twice the amount of chemotherapy to a tumor, you would get significantly better responses. And this was actually shown clinically by Dr. Bob Ozels in the 80s, to where he, did a clinical trial and just, I don't want to say overdose, but he gave super high doses of chemotherapy and was able to show responses because you got twice the amount of that therapy to the tumor. So I'm not suggesting patients undergo or go through that, but
Right.
what we wanted to show was, could we find a way to be able to deliver a lot more chemo or therapy to the tumor in a safer and more effective way? That means that we could essentially treat the patient and kind of make it more tolerable so the patients can actually have effective responses. They can get enough chemotherapy to where they actually see tumor regression. And so that's what
Mhm.
we did. We took the expanded porphyrin and we attached therapies to it. And we were able to show that in a lot of our clinically relevant early stage models, we saw tumor regression. So we saw some cures. We saw significant increase in overall survival in various solid tumors. We saw an increase of the therapy to the tumor relative to your standard of care stuff. So we did all the appropriate controls. And we did safety. We did some safety studies as well to show that we can dose so much higher with this expanded porphyrin with drug on it. We can dose so much higher relative to your standard chemotherapy control. In fact, at our high dose, the animals are totally fine. At that same dose with your normal kind of run of the mill FDA approved chemotherapy, it killed about 40% of the mice because it was so toxic. So I say that because it kind of illustrates this point that we have now something that is has higher tolerability. It is a lot more efficacious. You get a much greater response. We can overcome platinum resistance, which is a serious issue in a variety of different cancer types. I mean, this is a major, major problem. Um, and people are still getting that chemotherapy every single day. And clinicians cannot stand it. The reason why we have a scientific advisory board, the reason why we have the scientific advisory board that we do is because we have clinicians who understand that platinum is initially beneficial, but eventually it stops, and they really just want a safer, more effective platinum therapy, that they could treat the masses with. I mean, if you think about, okay, here's a little bit of business. If you think about the potential market that you could capture with a safer, more effective, platinum-based therapy, I mean, it's a massive market.
Mm-hmm.
And we understand that. We understand its potential. We have colorectal oncologists. We have ovarian or a gynecological oncologist. We have lung. We have all different, a variety of different oncologists on our scientific advisory board who have really expressed this immediate need for this type of treatment. And we're really the only ones that can do it. And I'll tell you why. And I know we're running out of time, so I would love to keep the conversation going. The reason why we're the only ones that can do it is because the type of payloads or therapies that we're delivering to tumors, they're completely incompatible with biologic medications, like antibodies. They're completely incompatible with other types of peptides or other types of delivery systems. They just can't do it. Actually, these biologics and peptides, they actually destroy platinum. And so it's we're the only ones that really, in this current market, in this current era, we're the only ones that have successfully found a way to effectively and selectively deliver platinum to solid tumors.
And I know you have to run in a second, but in short, where is it now? I mean, is it still in this research phase trying to get it to the market, or what's it look like now?
Right. Yeah. So we're not in research. We're in what we call development. So preclinical development. So we're embarking on in order to get into the clinic, you need to submit what they call an IND, or an investigational new drug application, to the FDA. And the IND consists of a variety of different safety studies and your narrative on how effective it is that are required for submission. And we're in the middle of that. So we're actually in the beginning stages of the IND enabling process. So we're about a year from the clinic. And we are hopeful that we will be able to submit an INT within that time frame to then initiate a phase 1 clinical trial in patients that are suffering from solid tumors, which is standard in the oncology space.
Yeah, and I wanted to, obviously I want to let you run away, but I wanted to first say I appreciate you coming on here and dishing out all of this information. I mean, we had a conversation about essentially the steps to being the cure to cancer with personal experience, obviously seeing the deterioration of the chemo take over the whole body and not be targeted.
Mm-hmm.
So uh, of course, I just wanted to say I appreciate you making the time and sitting down with me and chatting about this. I appreciate it.
Absolutely. Anytime you want to have another conversation, happy to do it.
Sounds good. I appreciate it. Thank you for taking the time out of your day to listen to our podcast. If you enjoyed this episode, please share it with friends, family, or anyone who could benefit. And please don't forget to subscribe and leave a review on your preferred platform. It helps us spread awareness, and we would be extremely grateful. You can also follow Rarely Discussed on Instagram, YouTube, as well as TikTok. If you're someone seeking diagnosis for porphyria, visit porphyriafoundation.org to receive a free doctor packet, forms for free DNA testing, and a list of doctors in your area. Stay tuned for more episodes of Rarely Discussed.